Well, allow me to introduce myself. I'm Don Stoltz, and this is what we're going to be looking at today, and this will probably be posted in the form of 2 or 3 presentations, which will be put up on Brightspace with voiceover, as I'm doing now. And they'll be labeled presentation 1, 2, and if necessary, 3. Viruses are inherently a little bit difficult to define. My definition you'll find on the next slide. I don't want you to memorize it, but I do want you to understand it. Understanding will come in large part by comparing them to cells. And the differences between viruses and cells are on the 3rd slide, which I think I'll move to the 2nd slide next. All right. There are a few interesting items here that you need to pay attention to. One is that all viruses, by definition, have a protein coat, so they have proteins. But, unlike cells, they don't have ribosomes, and therefore they cannot synthesize their own proteins, which is why all viruses are necessarily parasitic within cells, because cells have the ribosomes, which are the protein synthesizing machinery that is required for this particular component of a virus. The other thing I'll draw your attention to here is the way they replicate. And this is very different. In the case of viruses, you synthesize components and then particles are assembled. In the case of cells, you have a growth and division mode of replication. So there are a couple of conclusions you could draw from that. Well, one you might not, but anyway, if you look at a population of cells, you will see that they're not all the same size and shape, and you'll see that in the 4th slide. They can't be all the same size and shape because some of them are dividing, so they'll be different, and if they're growing, they'll be of different size. On the other hand, if you look at a population of a typical virus, this isn't true of all of them, but it's true of typical viruses, they're all going to look alike. And that's a consequence of the way in which they're put together. And you'll see examples of that in the 5th slide. So I'm going to move to slide 3 now. All right, so here we see the essential differences between viruses and cells, and I'll be talking about some of these items in much greater detail, focusing first on the protein, on the genome, and then on the protein coat, and then the envelopes. Draw your attention to the last item here. Cells, even though they grow and divide, so one will ultimately split into 2, these entities remain intact, right? So in other words, there aren't breaks in the plasma membrane, even when they divide. So the cells remain intact. Therefore, there's physical integrity maintained between the generations. That's not true of viruses. As we'll see later when we consider virus life cycles, viruses have to at least partially and usually completely disassemble after they infect a cell, or else their genome cannot access the cellular machinery required to make proteins and more genomes. Okay? This slide simply illustrates what I said before, that if you look at a population of living cells, they're not all going to look the same size. Upper left, we have bacteria. I mean, you'll see some are bigger than others. Some have spores and some do not. The other 2 slides are of yeast cells, and again you can see that they're not all the same. That's because they grow and divide. Here we have some examples of viruses, and I just wanted to draw your attention to the fact that in the upper 6 panels, you've got 6 different viruses, or different virus populations. And you can see that even though some of these particles are very complex, particularly the 2 on the right, they're all the same size and shape, right? And that is a consequence of how they are put together. We now come to the subject of viral genomes. And as you may recall from slide number 3, a viral genome can be just about anything, depending on the virus itself, whereas a cellular genome is always double-stranded DNA. As we're going to see in the next slide, viral genomes are of every variety. And here they are. So, unlike cells, viral genomes can consist of RNA instead of DNA, and it can be single-stranded instead of double-stranded. So again, it's always double-stranded DNA in the case of cellular genomes. Now, I think we'll move on to the next slide, because there's some really important things there, and you really kind of have to learn this slide and what's on it. Not necessarily the amino acid sequence, but all right, here's what we've got. This is often referred to as a standard mode of transferring information from DNA to protein. We now know, of course, that information can move in the opposite direction to some extent. Anyway, this is kind of the standard thing that happens in cells. Now, one thing that is really important here, and that's our cellular RNA pol, which means RNA polymerase, which is the enzyme that uses DNA as a template to make messenger RNA. Now, cellular RNA polymerases can only read DNA. They can't read RNA. So what's that mean? That means if you're an RNA virus, you're a virus with an RNA genome, you cannot use cellular RNA polymerase to make copies of yourself or to transcribe yourself. And for this reason, an RNA virus must encode, or the genome of an RNA virus must encode its own RNA polymerase. Right? Now, as you saw in the previous slide, in the case of single-stranded RNA viral genomes, there are 2 types, so-called plus sense and minus sense. A plus sense single-stranded RNA viral genome, if it gets into the cell, it can be recognized, it is recognized as messenger RNA, and the cellular ribosomes will translate it and make protein. One of the proteins made will be the viral RNA polymerase, right? Which then can make minus sense copies of the RNA genome and transcribe that to make more plus sense copies. Now, a minus sense RNA virus genome cannot be read by cellular ribosomes, and so cannot be translated, right? So that kind of virus always must bring with it into the cell 1 or more copies of its own RNA polymerase, because otherwise it's dead. So it brings its own RNA polymerase in. That RNA polymerase will transcribe it, make a plus sense copy, and then that can be translated by cellular ribosomes. Okay? What else do you need to know here? Okay, so in the case of molecules like DNA and proteins, there's polarity. So one end is different from the other. In the case of a single-stranded DNA strand, you have a so-called 5' end and a 3' end. And when you and this in double-stranded DNA is paired with a homologous copy which is anti-parallel. All right? Meaning that if it's double-stranded DNA, the 5' end of one strand will be paired with the 3' end of the other. The other thing to keep in mind is that transcription and replication of DNA occurs in the 5' to 3' direction, which means that nucleotides are added at the 3' end to it. So it appears to be, the molecule appears to be extending in the 3' direction or at the 3' end. You really should consult slides 34 and 36 to get a better sense of how this happens. Again, I'll draw your attention to the 4 bases that comprise a double-stranded, well, single-stranded or double-stranded molecule, and in the case of RNA, thymine is replaced by uracil. All right? So study this, learn it, see which nucleotides pair with which. That's important. This slide is simply a summation of what happens in the case of a plus-sense single-stranded RNA genome. TL means translation, TC means transcription. The plus-sense genome can be translated because it'll be recognized as messenger RNA, and then you get your proteins made. The minus-sense virus has to bring its own RNA polymerase into the cell, and then it can make the plus-sense copy, which can be translated. Okay? We now move to capsid structure. Now, here's the point. You go back to the slide which describes differences between viruses and cells. You'll see that most viruses, not all, but the vast majority, are very small, in the size range of 20 to 200 nanometers, most. So being small means they can't encapsulate or package much nucleic acid, right? Means typical viruses can't encode a huge number of proteins. So this means that they have to be economical in the use of what little genetic information they have. So ask yourself some simple questions. If you're a virus, a small virus, are you going to build your capsid out of several copies of different proteins? Not likely, because if you have different proteins, you're going to have to package a gene for each one of them. So most likely you're going to build your capsid, in the simplest cases, of many copies of one protein. Similarly, is that protein going to be large or small? I think you know the answer. It's going to be small, because then you need to package a small gene, not a big one. Now, what else is possible? So think about building a brick wall in your house. Are you going to use bricks of different sizes and shapes? No, you're not, because your wall wouldn't fit together very well. Are you going to assemble them haphazardly? No, because the thing's going to be unstable. You're going to put them together in a regular manner. What's the major expense? Labor. So if you really wanted to build a house efficiently, you design it in such a way that the bricks could assemble themselves. And this is exactly what the smallest viruses are able to do. In the next series of slides, we'll speak to that issue, I hope. All right. All right, so here we have two possible arrangements. And in the case of viruses, helical symmetry is by far the most common. And so these two diagrams kind of describe a helix. On the left, we have what's called a stacked disk configuration. The circles represent our identical polypeptides that we've been talking about that are coming together in a regular manner. And on the right, we have a helical version. The best way to describe this is to identify two neighboring polypeptides, in this case labeled A and B, and ask yourself how they are related to each other. And by that I mean is, how do you take A and put it on top of B, such that the overall pattern remains identical to what you started with. So in the case of the stacked disk configuration on the left, you can get A on top of B by a simple rotation. You just rotate A around the central axis, and when it's where B is, the thing is unchanged. On the right, if you were just to do a rotation of A, then A would be below B. It's not on top of B. So you can do a rotation and put it below B, and then a translation, a movement upward along a straight line, along the axis of the cylinder, and then A will be on top of B. So in other words, A is related to B by a rotation and a translation. That is how you define a helix. And we see examples, they're really, really, really common in nature in the next slide, but they're exceptionally common in the world of virology. All right, so here are three examples of helices in nature. There's a model of a viral nucleocapsid on the left. You can see that A is related to B by a rotation about the axis and a translation up along a line. Next we have an orchid, and you can see that each flower is related to its neighbor in exactly the same manner. This is a Nova Scotian orchid, by the way. And on the right we have a baculovirus capsid. If you look closely at the fine detail of this capsid, you will see lines crisscrossing it close together, and they are not at right angles with the central axis of the cylinder. They're at an angle. That is diagnostic of a helix. That's the kind of thing you would expect to see. Now, I said that there were two most common shapes for viral nucleocapsid. One being the cylinder, and it's usually a helical arrangement of proteins. The other is the icosahedron, which at low magnification, as we'll see in the next slide, kind of looks like a sphere, but it isn't. All right, so here we have a frog virus. This one replicates in the cytoplasm. It's actually a fairly large virus, about 130 nanometers. At this magnification, it looks kind of like a sphere, but if you go up in magnification, as we'll see in the next slide, it is not a sphere. Here's the same virus at a higher magnification. This is an icosahedron. Icosahedron. Google it. It is a solid. It has 20 sides, all of which are equilateral triangles. And if you slice this through a three-dimensional icosahedron, such that now you have two-dimensional slices, it's like cutting through a brick of cheese. I mean, you know what kinds of shapes you'll get. And by the way, if you cut the corner of a, start slicing the corner of a brick of cheese, you can get triangular slices. Okay. Anyway, you look closely here. You're going to see that there are pentagonal, 5-sided slices, fairly common, and hexagonal, 6-sided slices, and you can even see what look like small triangles here and there. You can only get these profiles by cutting through something which in 3 dimensions is an icosahedron. So this is the other of the 2 most common shapes of viral capsids. So now the question becomes, how do we fold up an array of identical polypeptides such that you get this kind of structure? And that's what the next few slides will speak to. All right, here we have 2 arrays of identical subunits. We'll call them identical polypeptides, as we did before. The upper array, of course, as you now should know, it can be folded up to form a cylinder in which every polypeptide is in the same environment, and we have said that should provide maximum stability. Can you fold that up to make an icosahedron such that every polypeptide is in the same environment? No, you cannot. But you can do it with the array shown below. This array is rather different from the one above. So in the one above, you have translational symmetry in the sense that you can take a polypeptide labeled A, which I've kind of shown on the right, and you can move it to position B, and the overall array has not changed. The structure of the array is the same. Now, you can do that in the lower diagram as well. You can move A to position B, and the overall arrangement of everything is the same. But you also have another symmetry operation you can carry out, and that's rotation. And I've identified axes of 6-fold rotational symmetry. Now, what is rotational symmetry? So a structure is said to have rotational symmetry if in a complete, which is to say a 360-degree revolution about the axis of symmetry, the overall structure of the thing is the same in 2 or more positions. So this particular structure, if we look at a group of 6 polypeptides, let's look at the group where 2 of them are labeled C and D. We could label the other 4 if you like. If you rotated C to put it on top of D, the structure looks the same. And if you kept going so that C was down in the position below D, the structure still looks the same. So in a 360-degree rotation, counting the starting position as number 1, there are 6 positions in that complete revolution where the structure is identical. And so that is that structure then, or that group of polypeptides in this case, has 6-fold rotational symmetry. This kind of array can be folded up to form an icosahedron, as we'll see in the next slide. All right. On the upper left is a bit of our array of identical polypeptides, and the array possesses both translational and rotational symmetry. Look at the gray sector there. It's a 60-degree sector. So imagine yourself with a pair of scissors. You can do this. And you're going to cut out that sector. And then you're going to pull the cut ends together. You could tape it together. And now what have you done? You have changed an local axis of 6-fold rotational symmetry into a local axis of 5-fold rotational symmetry. Plus, you've now built some 3-dimensional curvature into the overall structure. You convert 12 neighboring six-fold axes of rotational symmetry doing the same thing, 2 axes of five-fold rotational symmetry, and now you have an icosahedron in which every polypeptide is in the same environment. Right? And basically what you have here is a structure in which, again, the icosahedron has 20 sides. They're all equilateral triangles. And in the simplest case of a virus structure, you'll have 3 polypeptides per side. And since there's 20 sides, there will be 60 polypeptides. So you need 60 identical polypeptides to make the basic icosahedral design. Now, and this would be the smallest design. As you've already seen, some icosahedra are pretty big. The one I showed you before is 130 nanometers. So it's got more than the basic 60 polypeptides. And the diagram, the models on the lower left and the electron micrograph on the lower right will deal with that issue in part, and we'll consider that later on. This is just a larger example of the same kind of array. I mean, if you want to fool around with this, make a cylinder or an icosahedron. You can make some copies of it and get out your scissors and tape and see how you do. All right, I'm not covering the previous slide. You can ignore it. I did want to point out that while the icosahedron is one of the 2 most common shapes for viral capsids, a few of them are spherical. And you can actually tell that using an EM technique referred to as shadowing, an example of which is shown on the upper left. And you'll see the shadow denotes a spherical overall configuration. That's a rotavirus, by the way, but you don't have to know that. Upper right, you can see the shadows cast by an icosahedron. And they're angular, so they're totally different. You can actually prove that something is an icosahedron by the shape of its shadows. And that, in fact, is how the first icosahedral virus was identified as having that actual form. On the lower left is a geodesic dome model. And we don't have time to go into this, but the principles which underline the design of geodesic domes are exactly the same principles that apply to the construction of icosahedral viral capsids. Similarly, on the lower right, we see an actual, I think this is the U.S. pavilion at the 1967 centennial in Ottawa, or was it Montreal? I don't remember. No, yeah, I think it was Montreal. But anyway, this is a geodesic dome, and again, this can be described in mathematical terms using the same simple formula that you would use to describe the symmetry of an icosahedral virus. So some viruses have the form of geodesic domes rather than icosahedral, but the symmetry is the same. Okay, you can ignore slide 23. This slide is simply added to show you that some viruses have elements of both icosahedral symmetry and cylindrical symmetry. In this case, this is bacteriophage lambda, a very famous phage. The head has the shape of an icosahedron. And the tail is a cylinder, and I can't tell for sure whether it's a stacked disc, I think that's what it is, or in this case a helix. This slide shows a very rare example of a non-viral icosahedron occurring in nature. Typically, if you find something icosahedral in nature, it'll be a virus. So this is a carboxysome. It's a bacterial structure. I know some of you are wondering why virus capsids don't have the form of a cube, because in theory you can build a cube in such a way that all the polypeptides are in identical environments. There are reasons for, or at least speculative reasons, why no virus has ever been found to have the form of a cube, ever. So for one thing, think about how many polypeptides you need to build this structure. You've got 6 sides, and if they're all in the same environment, you will have 4 polypeptides per side, as I've shown here. So that's, what, 24 polypeptides versus 60 to build an icosahedron. What does that mean? It means for a capsid of the same size, if it's a cube, and you're going to cover all the surface, the polypeptides are going to have to be larger. Whereas in an icosahedron, since you've got 60 of them to cover the same surface, they can be smaller. So it makes sense in that way. The next slide kind of gets at another question, which is, what if you want to take your basic design and enlarge it? Which you would do, again, ideally using more copies of the same polypeptide. So I'll just put that slide on and very quickly go through what results if you try to do that. All right, so on the left we've increased the design. So now we have what, way more polypeptides. But look what's happened. They're not in the same environment now. You'll see some polypeptides, those on one side, for example, they're interacting with each other on a plane, right, on a flat surface. But if you look at 2 that are interacting over the edge, where 2 sides come together, you'll see they're interacting at a 90-degree angle. It's a lot to expect a polypeptide to be able to function in 2 completely different environments. Whereas if you try to enlarge the basic icosahedral design by adding more triangles to each side, as I've done on the right, if you look at the same thing there, you'll see that the polypeptides are not all in the same environment, but the difference of the environment is not huge. And so you can envision that the same polypeptide might actually be able to accommodate 2 slightly different environments, and that's probably what's going on. This brings us back to this slide, which we have encountered before, and it brings us to these models on the lower left. We saw that in the simplest design, upper right, you have 12 axes of 5-fold rotational symmetry. If the 5 polypeptides at those corners or vertices were clumped really closely together, they would look like knobs, each one of which is surrounded by 5 others, as you can see. That's the model on the lower left. Each clump, they're called capsimers, is surrounded by 5 others. Other models, you can see that yes, there are 12 capsimers surrounded by 5 others, but there are now others that are surrounded by 6 others. And if you look closely at the electron micrograph on the lower right, you can see capsimers surrounded by 6 others. How do you pull that off? Well, the way it's done is that instead of converting adjacent axes of local 6-fold rotational symmetry to axes of 5-fold rotational symmetry, you spread them out. So for example, in the array at the top left, you've cut out one 6-fold chunk, move to the right and leave that 6-fold chunk in, and then keep moving to the right and take out another 6-fold chunk. You're going to end up with the model shown in the middle bottom. of that group of 5 models. Okay, so there's a way to do it. Let's look at the next slide. So here's an example of that sort of thing. This is a human adenovirus, and if you look at the inset on the upper left, you can identify capsimers, or clumps of identical polypeptides surrounded by 5 others, and all the rest are surrounded by 6 others. Okay? These capsimers are all made of identical polypeptides. Okay, we're going to finish this presentation by looking at viral envelopes. An envelope, by definition, is a unit membrane situated exterior to the nucleocapsid. Mostly, in most cases where viruses have envelopes, they are acquired by a process called budding. Budding refers to the acquisition of a unit membrane by a viral nucleocapsid pushing through a pre-existing host cell membrane. Usually, but not always, it's the plasma membrane. As we see in the upper 2 examples and the middle left, but the middle right, it's pushing through the endoplasmic reticulum and acquiring an envelope in that manner. On the lower left, you've got a virus actually budding through the nuclear envelope. That's really complicated, but we're going to get into that more when we talk about herpes viruses, which we're not going to focus on in this. Well, not in this particular presentation, but I think in the next one. Lower right is not an example of budding, because the membranes that are surrounding the nucleocapsids, which are cylindrical in this case, of course, these membranes, if you look at them carefully, are open-ended profiles. They have no continuity with pre-existing cellular membranes. So these envelopes are being acquired de novo, from scratch. Nobody knows how this happens, but it does. And with that, I think we'll end this first presentation.